Alright, just need to check that this is streaming okay, so bear with me. Hello, hello, hello. Test, test, test. Cool, okay, that's working. Nice. So, I want to... I want to do a deep dive on DNA sequencing. So I want to be able to achieve something at the end of this, um, like discovery of how things work. So I'm going to lay out a plan, but first I'm going to do a Duolingo lesson. So let's see how I go. Work me into actually wanting to do anything. Does she love me? Rakastaako. I guess it's that.
Rakastaako
Does she? Han
hän
sinua?
sinua?
Something like that. Han minua. Oh, me. Yeah. Okay, that makes
Onko
sense.
sinulla poikaystävää? Sinulla.
Onko sinua? So does. Oh wait, what's that one?
Poikaystävää.
Boyfriend. So does he have a boyfriend? Onko sinulla? Do you? Okay. Sinulla, you have. That's right, because it'd be Hanella otherwise. Do you have?
Onko
Onko, do you have?
sinulla
Sinulla, a girlfriend? Tut
tyttöystävää?
Tyttöystävä, I think it's that. Do you have a girlfriend? Yeah,
Onko
nice.
hänellä poikaystävää?
Does he have a boyfriend? Darling. Darling was Kalta? Maybe? I think it's Kalta. Yeah, okay.
Kulta.
I do not want, I do not want.
En
En
halua
halua
sinua.
sinua angry. Vihainen? Okay.
Vihainen.
So, etta ole. So, darling, I do not want. En halua. Okay. You. En halua. To be angry. To be angry. Etta olet. Interesting. I don't remember what
Että
etta is. You to be. Etta olet. Okay, cool.
olet vihainen.
Nice. Kalta en halua etta olet vihainen. Alright, cool.
Kulta, minä en halua, että sinä itket.
Darling, I do not want you to be. I do not want you to cry. Darling, I do not want you to
Sinä.
be crying. You to cry. Okay, fair enough. Cool. Rakistan, I love. I think it's that. This is this. So I love this. Poroista would be. I can't even remember. Dog. Poroista dog. Can't remember. Maybe it's that. Ah, I just feel it's that. This fluffy poroista. Oh yes, that's remember. I remember that one. Cool. Can you actually see the screen? Kinda. You can kinda see the screen. And also, I just realized my awesome image is gone. Where is my awesome image? Let me just add my awesome image. Oh yeah, it's all about the image. Cool. She really loves this horse. Oh, I also need to change what I'm streaming now. So accept, accept. It's not a game, it's design or creative, other. Maybe just nothing. Can I do that? Yeah. Alright, cool. She really loves this horse. Han Todella loves Rakasta? Something like that. Rakasta tata hevosta. Rakasta. I can't remember why. I love you, I guess. Rakastan Sinua. Otsu, I love you. Otsu Mina, I love you. So maybe Rakasta? Rakastan. I love. Fair enough. Makes sense. Humuilen Koskahan Rakasta. Humuilen is I'm smiling because she loves me. Johnny Onko Sinula girlfriend Tutto something. Tutto Ustava girlfriend. Oh yeah, okay. Tutto Ustava. Is that one? Yeah. Girlfriend. Nice, that makes a lot of sense. Or boyfriend. Ustava.
Rakastatko sinä tätä poikaa?
Rakastatko sinu... What did you say?
Rakastatko sinä tätä poikaa?
So, does the boy love you or something like that? Tata, I can't remember. Do you love this boy? Do you love Tata is this boy? Okay,
Onko
cool.
hänellä tyttöystävää?
Whoops. Onko hänellä tyttöystävää?
Onko hänellä tyttöystävää?
Onko hänellä tyttöystävää? Okay, cool. Does she have a girlfriend?
Kulta, haluan, että sinulla on hauskaa.
Whoa, whoa, whoa.
Kulta, haluan, että sinulla on hauskaa.
Darling, I want you to have... I want you to laugh or something like that. I want you to laugh. House girl. I want you to have fun. I see, yeah. Laugh is not... Should be fun. Cool.
Onko hänellä poikaystävää?
So this is... Do you have a... Well, does she have a boyfriend? Poikastava. Yeah.
Minä rakastan sinua.
Rakastan sinua. Rakastan sinua. I love you.
Hymyilen, koska hän rakastaa minua.
Humuilen. I can't actually remember how this spells.
Hymyilen, koska hän rakastaa minua.
So, I'm smiling because he loves me. Does she love me?
Rakastaako
hän sinua? Does she love me?
Minua. Onko sinulla poikaystävää?
Do you have a boyfriend? Alright, cool. Good progress. Okay, so this plan, let's do this. Go away. Close. Alright, cool. So I've got a few things I need to know. So how do you break up a DNA sequence? I've heard about CRISPR. How do you put the DNA sequence back together? I've heard about some natural process in the body that does this. Like if you take one part of the DNA strand out, like it always comes in a pair, and so your body is trying to look for the part that you took out. And then so when you present it, you can actually insert extra information, but as long as it still thinks that it's the same, like I guess connectors, we're going to find this out, then it assimilates it, it takes the position, and they use this to identify the pathing of genetic disease. So they add like a signature to the DNA that they put back in, and then that allows them to kind of study what was affected. So I think something like that. I don't know how to word it. I don't know what the name is. So we need to know what that is. How do you put the DNA sequence back together? Um, we know about the body using its DNA pairs. Um, wait, we know something about where we know something about the body using symmetry in DNA, so that when I don't know, we'll call it side, one side of the DNA is corrupt, is that how you spell corrupt? Then it will hunt out a replacement. I wonder if it generates or generates a replacement. Um, in this case, you can inject sequence code in, um, and it doesn't know anything about you hijacking it. Something like that. It's a bit long, but hey, whatever. So that's really important because this is the whole premise of what I want to do. I need to know a little bit about like chemical structuring symbols. So when you have those like hexagonal lines and stuff, I want to be able to read them and feel like a deep connection to them. So I need to learn about what the symbols mean in chemistry. Um, that's fine. Chemistry and biology. I also want to know, so what kind of research have people done in terms of trying to engineer programs into DNA? Because this is also really crucial. Do I have to start from scratch and figure out myself, or can I start to read some white papers? I know I have been seeing quite a lot coming through from Manolis Kellis about all of the work his department are doing in MIT. So something like that. So find a good source of white papers, not heat papers. Find a good source of white papers um, regarding engineering of DNA sequencing, regarding engineering of DNA sequencing and manipulation slash hijacking. There was one more thing. I just gotta try and remember what it was. Um, oh, can you even see this? I bet you can't. There you go. That's probably better. Not that you need to see it. Can I mute my mic for a sec whilst I cough? Okay, cool. I got it. Um, yeah, so there was one more thing. Oh yeah, that's right. Um, what kind of machines are there that can automate testing? So I imagine, let's imagine that um I come up with some ideas for how a DNA sequencing might work, and I want to try it out. What kind of tools can I use to like link up, maybe use an API to automate some of that testing? You know, like a machine comes in and it fills a pipetting. So the pipette fills a little dish and um with a set of chemicals or whatever to join or disconnect or add up together again. And I want this kind of all driven from a computer. So so I can really go mad with the testing. So, what kind of machines are there that can automate testing? So I guess I want prices, pipetting or automated pipetting. Um, what does the what are the logistics around supplying um, with more uh, materials for the processes, API, that kind of thing. I think that's enough. I think when I start diving in, this will become a bit more apparent what I need. Yeah, I think that's it for now. Cool. Okay, so the plan is to essentially create um, like if you were to say you have a robot that I don't know the robot does your washing or something like that. I want to see or work towards. I want to work towards making biology that does that. So, um, I mean, I wouldn't say epigenetics, but I would say something like, what kind of process do we have to go through to to so is it possible to even do that kind of thing is number one, and number two is how hard is it to do that kind of thing. Like I imagine most of the scientists in this kind of part are doing computational sciences around DNA sequencing, so I imagine this is well and truly underway, but why are we not seeing more progress done? And I was hearing some news from George Hotz, I think, George Hotz, about how very slow it is to do or like biology at the moment isn't very particularly uh, well automated. So if I could come up with processes that automate some of this stuff as a kind of engineer, a developer, let's say, maybe that's going to give me like an edge, or I can help out the industry figure out things with the goal of not just creating engineered products like that you grow in a bottle and they become your dishwasher, but maybe we can interfere with some of the coding of your genes to, I don't know, eradicate disease, um, maybe regrow bones quicker or stronger. You know, I had this idea of having an exoskeleton company, and I just find that with things like Neuralink on their way, I'm not so sure the body is going to be that important anymore. You know, it's just a sack of flesh, and really the majority of the stuff, if not all the stuff, is happening in the brain. So, do we really need to care about fixing someone's ailments if you can just, visualize stuff? So here's the thing: the actual final goal is, can I create something that can improve the brain's bandwidth by figuring out DNA sequencing, um, I don't know, create tests for when you go to Mars or something, and you want to see if the biological human can deal with things. Can you engineer, like, I don't know, from a 3D model or something, and then send that up and see how it deteriorates in space and all that stuff. Um, Loads of ethical questions someone's got to think out. But yeah, let's crack that. So that's the kind of idea. So I'm gonna set a timer for. I say I feel a bit tired, so I'm gonna set a timer for like 2 hours, I think, and we'll see where we go from there, and just see how much information I can learn. So bear with me. Hey Google, set a timer for 2 hours. Cool. Okay. Right. Let's start. So first things first. CRISPR. Let's read this shit. Oh, I need to change the name as well. DNA sequencing. DNA sequencing deep dive. It'd be good if I could actually spell as well. I'm pretty sure that's how you spell that. Gene-based medicines, transforming the lives of patients with serious disease. Download CRISPR Therapeutics corporate presentation. Okay, that sounds good. Programs, hemoglobinopathies, however you say that. Let's just get a feel. Something to do with blood. Ah, failed. Absolutely failed. Oh, whatever the matter. Doesn't matter. Globulin, hemoglobulin, pathological stuff, I don't know. Aiming to treat beta thalassemia and sickle cell disease with gene editing, hematopoietic, something stem cells. Okay, cool. Immuno-oncology, creating the next generation of cell therapies for cancer enabled by gene editing. In vivo, editing cells inside the body to treat genetically defined diseases. Yeah, this is it, editing cells inside the body to treat genetically defined diseases. Next generation of therapies for cancer enabled by gene editing. Yep. What is this? Thalassemia is the name for a group of inherited conditions that affect a substance in the blood called hemoglobin. People with thalassemia produce either no... Ah! Go away, stupid things. Produce either no... Oh, here it is. Produce either no or too little hemoglobin, which is used by red blood cells to carry oxygen around the body. This can make them very anemic. Cool. Nice. Understood. And then... I hate these websites. Broadening the applications of stem cells through gene editing. Okay, let's get a feel for this website first before I click through too much. CRISPR-Cas9. Can you actually see this stuff? Let's try that. And they keep buzzing in my pocket as well. Okay, work people. Rapidly translating a revolutionary technology into transformative therapies. Cool, let's see what they got.
Today, most medical therapies aim to treat diseases by targeting abnormal proteins associated with a disease. Usually, these abnormal proteins exist because of mutations in specific genes in a patient's DNA. In the past, we have not been able to alter these genes directly to address the root cause of a disease. However, a novel gene editing system known as CRISPR-Cas9 may allow us to modify, delete, or correct specific areas of our DNA in order to treat these diseases. The system is comprised of two parts: Cas9, an enzyme that cuts DNA, and a guide RNA whose sequence.
So, Cas9 enzyme. Oh fuck, I can't even remember what they said.
An enzyme that cuts DNA.
Enzyme that cuts DNA.
And a guide RNA whose sequence directs Cas9 to a specific location in the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that can be easily the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that is comprised of two parts: Cas9, an enzyme that cuts DNA, and a guide RNA whose sequence directs Cas9 to a specific location in the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that can be easily and precisely targeted to a desired site in the DNA. The CRISPR-Cas9 gene editing process begins when the complex recognizes and binds to a short segment of DNA adjacent to the target site. This initiates unwinding of the DNA helix, which allows the guide RNA to pair with a specific target sequence in the DNA. If the sequence is
Right, this simulation, that's going to be really helpful. I wonder if you've got some tooling for that as well. So let me add to my little thing here. Is there simulation tooling to save? Well, yeah, is there simulation tooling to save me some money on trying to get the kit? I know they're quite expensive, these machines that do like automated pipetting, like 5k or 2k to like 15 grand. So and plus, I'm sure. So if I can get some tooling that is already made to kind of play around with this stuff, it might even be cheaper, although it's probably licensed. So let's see, is there tooling licenses for the simulation stuff that's affordable? Excuse me.
pair precisely, Cas9 cuts the DNA, forming a double-strand break. Our cells respond to such breaks by activating natural DNA repair pathways. In some cases, a process called non-homologous end joining results in the addition or deletion of a few base pairs, which disrupts the original DNA sequence and can cause gene inactivation. A larger fragment of DNA can also be removed by using two different guide RNAs to target separate sites on either side of the desired deletion. Cleavage occurs at each site, and the repair process joins the separate ends, thereby deleting
understand that.
which disrupts the original DNA sequence and can cause gene inactivation. A larger fragment of DNA can also be removed by using two different guide RNAs to target separate sites on either side of the desired deletion. Cleavage occurs at each site, and the repair process joins the separate ends, thereby deleting the intervening sequence. Corrections to DNA can also be made by adding a DNA template along with a Cas9 guide RNA complex. The template is designed with sequences.
Cas9 RNA guide complex. Let's put this under a new section, maybe something like search keywords. Ah, God, I can't type. Search keywords. That sounds good. Hey, this does sound like what I've heard, so that's good to know that I'm on the right track. The deletion of both the sides with some targeting, that's really interesting. The fact that the guide RNA this can target specific things. I wonder what the extent of that is. If you could, like, can you target entire frameworks of DNA sequences? How hard is it to engineer that targeting? Can you engineer the targeting, or is this RNA guide RNA, is that something that the body makes, or is this part of the CRISPR thing, which is that you have some ability to engineer things? What are the tools that you use to engineer them? Is there like a programming language for this, or is it much more like manual, physical labor of trying to get it all done? Are there some shortcuts I can take? Like if I wonder what kind of data sets I can get hold of that allow me to see what's already been done and engineered. I might be able to run some AI over it to try and learn some patterns, see what happens. If I could generate pipetting using the AI, we could just see what actually occurs. Yeah, interesting. So I see. So they're kind of going through the list of things that it does. Disruptions, deletions, corrections. Disruptions is interesting. I'd like to know, because I'm type 1 diabetic, I'd like to know the kind of the DNA relevant stuff for that disease, see how much they know, because maybe there's some like AI driven solution to that problem as well that I can start to code together. Yeah, interesting
that
anyway.
exactly match the DNA adjacent to the target cut site. Through a process called homology-directed repair, the cell uses the template to repair the break, thereby replacing the faulty DNA.
Homology. I know the word actually. Just gotta remind myself what it is. Mathematics. Homology is a general way of associating a sequence of algebraic objects, such as abelian groups or modules, to other mathematical objects such as topological spaces. Homology groups were originally defined in algebraic topology. Let's see. Sometimes they have quite different meanings. Homology, biology. Is homology is similarity due to wait. What's the overall one? The state of having the same or similar relation, relative position or structure. I did know that. Okay. Is similarity due to shared ancestry between a pair of structures or genes in different taxa. Right, well, cool. It's got little. I didn't know Wiki did that. That's amazing. In biology, a taxon is a group of one or more populations of an organism or organisms, seen by taxonomists to form a unit. Although neither is required, a taxon is usually known by a particular name and given a particular ranking, especially if and when it is accepted or becomes established. Pair of structures or genes in different taxa. A common example of homologous structures is the forelimbs of vertebrates, where the wings of bats and birds, the arms of primates, the front flippers of whales, and the forelegs of four-legged vertebrates, like dogs and crocodiles, are all delivered from the same ancestral tetrapod structure. Cool. Oh, my boy Charles there. Let's go. Evolutionary biology explains homologous structures adapted to different purposes as the result of descent with modification from a common ancestor. The term was first applied to biology in a non-evolutionary context by an anatomist, Richard Owen, in 1843. Homology was later explained by Charles Darwin's theory of evolution in 1859. Being observed before this, Aristotle onwards, and it was explicitly analysed by Pierre Bellon in 1555. Who cares? Great. Good for you guys. Let's do this. So, let's continue. Just going to rewind a bit actually.
Exactly match the DNA adjacent to the target cut site. Through a process called homology-directed repair, the cell uses the template to repair the break, thereby replacing the faulty.
Yeah, that's exactly what I was talking about. Now I understand. So homology repair is this this kind of bilationship between the strand that you remove and the strand that's still there, so then it can use its own processes to correct itself. Um, naively, ignorantly, doesn't know that it's doing it. Hacked, hijacked.
DNA sequence, or even inserting a new gene.
Oh, can I have not more?
The break, thereby replacing the faulty DNA sequence or even inserting a new gene.
Damn. Okay, that's good to know. Um, Fine. Let's go and see this PowerPoint presentation. Creating transformative gene-based medicines for serious diseases. The present. Can you even see this? Probably not. Not that anyone's watching. The presentation and other related materials may contain a number of forward-looking statements within the meaning of the blah blah blah, yada yada yada. Leading gene editing company focused on translating blah blah blah. Specific, efficient, and versatile tool for editing genes. Delete, correct, or insert, disrupt. Okay. Our pipeline. Okay, program. CTX001 trademarked. Ooh, yeah. Research, industry enabling, clinical, marketed. Sickle cell disease. So what are the, let's have a look at these things, what these actually, I'll get through this document first, but I'd like to know what the kind of, how they describe these things. Hemoglobinopathies, I don't know, devastating blood diseases. Yeah, upregulating fetal hemoglobin. Yeah, pioneering CRISPR trials. Phase 1 out of 2, international, multi-center, open-label, single-arm studies to assess the safety and efficacy of CTX001 in patients with transfusion-dependent beta thalassemia. Design target enrollment: 45 patients aged 12 to 35 years with TDT, including genotypes defined as a history of at least. Is this superscript? I can't remember the wording for this. Anyway, defined as a history of at least 100 ml kilograms year or 10 units year PRBC. Let's see what that is. Packed red blood cells. Gotcha. Pioneering CRISPR trials. So this is stuff that they've done already with it. Proportion. CRISPR trials, yeah. Proportion of patients achieving sustained transfusion reduction of 50% for at least 6 months. Cool. Primary endpoint. Patient baseline and treatment characteristics. Okay. Well. Nice. Allogeneic. Can you still hear me? Yes, you can. Cool. Wait. Allogeneic CAR T therapy has transformative potential. CRT. CART, what's that? Highly complex inhibitor is a type of immunotherapy which involves collecting and using the patient's own immune cells to treat the condition. What does it stand for? Chimeric antigen receptor. Okay, cool. Apheresis. God. Boring. Change CRISPR edited allogenic CAR T. Boring. Ah, actually. Improve persistence in the allo setting via beta 2M knockout to eliminate MHC1 expression. Ah, now we're getting into the details. Here we go. CRISPR gene editing in one step, designed to improve persistence in the allo setting. Don't know what that is. Allo. I guess it's saying, okay, combining form. Improve persistence in the allo setting via beta 2M knockout. What's beta 2M? Beta 2M must be a position in the DNA. Let's have a look. Beta 2M. Beta 2 microglobulin, also known as B2M, is a component of MHC class 1 molecules. MHC was shit. I just googled that, didn't I think I did. MHS, jeez. No, I did not. Major histocompatibility complex. The major histocompatibility complex is a large locus on vertebrate DNA containing a set of closely linked polymorphic genes that code for cell surface proteins, essential for the adaptive immune system. Okay, cool. So they improve persistence in the allo setting via that knockout to eliminate MHC expression, avoid need for more toxic lymphodepletion regimes, prevent GVHD via TCR disruption. TCR. TCR, T cell receptor, is a protein complex found on the surface of T cells or T lymphocytes. it remain lymphocytes. Oh, I hate it when that happens. Lymphocytes. Alright, well, fine. I'll just read you. Found in the T lymphocytes that is responsible for recognizing fragments of antigen as peptides bound to major histocompatibility complex MHC molecules. The binding between TCR and antigen peptides is of relatively low affinity and is degenerate. That is, many TCRs recognize the same antigen peptide. Wait, the binding between TCR and antigen peptides is of relatively low affinity and is degenerate. That is, many TCRs recognize the same antigen peptide, and many antigen peptides are recognized by the same TCR. T-cell receptor. This seems really relevant. The T-cell receptor is a protein complex found on the surface of T cells. Immune system again. Okay, fine. Let's just move on for now. Improves consistency and safety by precise insertion of CAR construct into track locus without using lentivirus or retrovirus. Track locus. Track protein. I swear I just looked this up. Track gene. Yeah, T cell receptor alpha constant. T cell receptor alpha constant. Okay, fine. Carbon trial design. Single arm study evaluating the safety and efficacy of CTEX. Blah, blah, blah. Patient flow and baseline characteristics. Does dependent responses observe? Dose dependent responses observed with CTX 110. We've got still acceptably safety profile with. Yeah. Nope, don't care. Enables regenerative medicine too. Okay. Medicine 2.0. Yeah, this is exactly what I'm thinking of. CRISPR Cas9 technology opens broader applications for regenerative medicine. Let me just see what's going on here. Opens broad applications for regenerative medicine. CRISPR-Cas9 allow immune evasion, improve cell function, direct cell fate, therapeutic targets, diabetes, stem cell technology, blah, 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 blah. Marketing, aim to develop beta cell replacement product to treat diabetes that does not require immunosuppression, applies immune evasive gene editing expertise from allo CAR T. I can't remember allo again. I know I googled it. Something combined, define, allo, not allow, that one. Okay. I mean, you could have just put that word, but anyway. From our different CAR T programs to stem cells. Plan to initiate trials in 2021. Oh, that's really interesting. ViaCyte. Companies of interest. ViaCyte. Okay. Diabetes. Therapeutic targets. In vivo applications of CRISPR-Cas9. AAV vectors for neuromuscular indications. Neuromuscular. Okay. Adeno-associated virus AAV to deliver Cas9 GR9 GRNA. I wonder what the G is. Guide RNA. Ha ha. Okay. Guide RNA. Nice. To muscle, the nervous system, and other tissues. Adeno-associated virus to deliver the Cas9 gRNA to muscle, the nervous system, and other tissues. Ah, this is it. This is it. This is what I wanted. In vivo applications of CRISPR-Cas9. This is the one. AAV vectors for neuromuscular applications. AAV vector. Oh, that's interesting. Adeno-associated virus. Vector, I like that word. Whoa. So they use this adeno-associated virus to deliver targeted manipulations. Wait, they use the virus to deliver the guiding RNA to use the enzyme to break it down. So they can guide you right into the problem with the virus. Okay, so we need to start thinking about, do we need to engineer viruses? Jeez. Let's do a different section for this. Plan. Oh, I don't like that. That's gross. Yeah, let's. Oh God. The plan. So I want to do what we found out. You can use adeno-associated virus to deliver. Oh, why am I doing that? That. Yeep. Okay. Collaboration with Stride Bio to improve tissue specificity. Ah, that's a word. I need to know that word. The quality of belonging or relating uniquely to a particular subject. The narrowness of the range of substances with which an antibody or other agent acts or is effective. The narrowness of the range of substances with which an antibody... Oh, okay, that's really cool. So collaboration with Stride Bio to improve tissue specificity and reduce immunogenicity. Is the ability of foreign substance such as an antigen to provoke an immune response. Okay, let me just read this. I think I might have misunderstood what this is. Unlocking in vivo applications of CRISPR. Associated virus to deliver to muscle. I don't know what this is trying to tell me now. Any clever people know? I guess no one's watching. Let me see. Maybe there is. Yeah, reach out if you know anything I don't. Programs include DMD and DM1 in collaboration with Vertex as well as other early research programs. DMD? What's that? Duchenne muscular dystrophy. Okay, gotcha. I knew that. Lipid nanoparticles containing with mRNA, encoding Cas9 and gRNA for delivery to liver. Lipid technology from MIT. My boys at MIT. CureVac. Lipid technology from MIT and mRNA technology from CureVac. Lipid is fat, isn't it? Some kind of fat delivery system. Any class of organic compounds that are fatty acids or their derivatives and are insoluble in water but soluble in organic solvents. Yeah. Soluble in water. What was it again? Insoluble in water but soluble in organic solvents. Like chemicals, I guess. They include many natural oils, waxes, and steroids. Oh, okay, cool. This is a really interesting slide. I'm just gonna make sure I've got this copied somewhere. Um, Optimizing the CRISPR platform, nuclease engineering, enhance CRISPR-Cas9 system through protein engineering. I like the scissors. We're gonna work with the scissors. Guide RNA optimization. Identify optimal guide RNA formats and sequences for therapeutic editing. Identify optimal guide RNA formats and sequences for therapeutic editing. I wonder what tools they have for this because I feel like this is what I need to understand what they're using first themselves as a company, and then see how I can kind of fit my APIs into it, or my requests to their APIs. How much engineering do I have to do myself before I can start trying to apply some AI onto my problems? Advanced editing, improve efficiency of gene correction and multiplexing. Synthetic biology, engineer improved cellular therapeutics. Yep. Ooh, I don't like this word. Um, I'm just gonna ignore it. Let's just ignore things I don't like. Okay. Cool. That's awesome. That was really helpful actually. Good overview. Let's see about us: mission, leadership, partnerships. Let's see who's in charge of this stuff. Samart Kulkarni, Chief Executive Officer. Steve Cafe. Where's the guys who actually do stuff? Head of Research and Development, Dr. Tony Ho. Let's have a look. R&D, Research and Development, blah, blah, blah. Tony Ho had a number of roles at AstraZeneca. Ooh, my boy. Most recently, Senior Vice President and Head of Oncology Integration and Innovation. PARP. PARP. Oh, we need to know these if we're going to talk to him. God. Oral medication for certain types of advanced cancer. Nice. PARP. Define PARP inhibitor. Dictionary of cancer terms. PARP inhibitors are a group of pharmacological inhibitors of the enzyme poly-ADP ribose polymerase. They're developed for multiple indications, including the treatment of heritable cancers. Wow, okay. Let's have a look at this. This diagram looks awesome. Parpy. Come on. You're not even trying now. Um, Wait. I didn't quite answer this. It's the cell cycle arrest. Something. I don't know what these are. About we and PARP. Let's have a look at we and PARP is. We1. Nuclear kinase belongs to the family of protein kinases. Jesus Christ. This is like learning maths. God. In biochemistry, a kinase is an enzyme that catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrates. Kinase. We should really note this down. Important. Oh, search keywords. I think I called it. Search keywords. Where did I put the rest of that? I put it somewhere. There it is. Let's move it from here. Let's put a little title here. Generic, I don't know, bio words. Kinase. Sounds important. Catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrates. We. Yeah, is a nuclear kinase belonging to the. Wait. We is a nuclear kinase belonging to the SERTHR family of protein kinases. So a kinase is an enzyme that catalyzes the transfer of phosphate groups from high energy phosphate donating molecules to specific substrates. Okay. In the fission yeast, Schizosaccharomyces pomb. Pomb. This is very specific. Okay, wait, is this specific? It's in the fission yeast's molecular mass of 96 and is a key regulator of cell cycle progression. Influences cell size by inhibiting the entry into mitosis through inhibiting CDK1. I'm starting to just I'll come up with a plan. So these kind of things, I wonder if we can abstract away some of the details here. So if I were to create a tool where I want people to be able to just like go wild, be designers, be gods kind of thing, I need to get rid of some of this, like, I guess Greek Latin-y kind of naming of things. We need something that's more like functionality. What does this do? Um, I bet there's a database. So let me see if we can. Is there a database of cool words that that instead show the function, um, that instead are named in a functional form rather than a specific form? Like, if you just had a list of these things and you're like, oh, I need to do this, and you just have this list of things, you could just have like a dictionary attack in it when you're trying to do your AI, and then it just finds the one that works. Something like that. Okay. Cool. So where was I? Let me just close some of these tabs down now. Homology, the state of having the same or similar relation. PR, BC, packed red blood cells. CAR T is chimeric antigen receptor. Allo was different. Beta 2 microglobin. Major histocompatibility complex. T cell receptor was what? I can't remember now. Undo close tab. T cell receptor is a protein complex found on the surface of T cells or T lymphocytes that is responsible for recognizing fragments of antigen as peptides bound to major histocompatibility complex molecules. The binding between TSR, T cell receptor, TCR, and antigen peptides is of a relatively low affinity and is degenerate. That is, many T cell receptors recognize the same antigen peptide, and many antigen peptides are recognized by the same T cell receptor. Yeah, okay, understood. T-RAC, the T cell receptor alpha constant, whatever that is. Now this is great. Gene cards, the human gene database. This is awesome. What is this? I like this. Looks like it's got shitloads of information. Protein products, antibody products. Whoa. Cytogenetic band. Track gene and genomic locations. Bands according to ensemble. So this site looks really cool. Let me just try and read one of these because I want to kind of get a hint of what kind of stuff they're saying. T cell receptors recognize foreign antigens which have been processed as small peptides and bound to major histocompatibility complex molecules. MHC. MHC is histo. Can't remember. What's MHC again? I literally just read it. It's so annoying. The brain is so dumb. Major histocompatibility complex. Large locus on vertebrate DNA containing a set of closely linked polymorphic genes that code for cell surface proteins essential for the adaptive immune system. MHC, immune system. MHC, major histocompatibility complex. Let me read that again. I've lost it now. Oh, here it is. MHC. So major. Oh, it says it here. I'm an idiot. Sorry. Major histocompatibility complex. So T cell receptors recognize foreign antigens which have been processed as small peptides and bound to major histocompatibility complex. This is awesome that they do that. Molecules at the surface of antigen-presenting cells. Each T cell receptor is a dimer consisting an oligomer consisting of two monomers joined by bonds that can be either strong or weak. Okay. Consisting of one alpha and one beta chain, or one delta and one gamma chain. In a single cell, the T cell receptor loci are rearranged and expressed in the order delta, gamma, beta, and alpha. If both delta and gamma rearrangements produce functional chains, the cell expresses delta and gamma. If not, the cell proceeds to rearrange the beta and alpha loci. This region represents the germline organization of the T cell receptor, alpha and delta loci. During T cell development, the delta chain is synthesized by a recombination. During T cell development, the changed chain is synthesized by a recombination event at the DNA level, joining a D segment with a J segment. A V segment is then joined to the DJ gene. The alpha chain is synthesized by recombination, joining a single V segment with a J segment. Okay. Oh, this is really tech stuff. I like this a lot. Let's get a little link on this one. Um, helpful pages, links. I like this a lot. What's the search like? So if I type in, what was that other one? It said DX001 or something. I can't remember. Is it in here? I think it is. Yeah, here it is. CTX001. So let's go and plop that in here. See what kind of thing comes up. No, okay. Let's look for I don't know, COVID-19 DNA, I don't know, segments. Number. Signature. Let's see if that comes up with anything. Okay. So what are these? Symbols, aliases, alias identifiers. What is this site called? Genecards.org. Okay, cool. I like it. I like it a lot. Let's keep that one open. Allo, different. Okay. Guide RNA, gRNA. Gotcha. Specificity. Narrowness. Yes, got it. Immunogenicity. Define. I don't care about COVID. What is this? Is the ability of a foreign substance, such as an antigen, to provoke an immune response in the body of a human or or other animal. Immunogenicity is the ability of a foreign substance, such as an antigen, to provoke an immune response in the body of a human or other animal. Okay. That's a disease. Lipid was that fatty acid. The PARP, that was part of his work. Yep. We1 was something very specific to something. It was some like protein. It's a nuclear kinase. Oh, okay, yeah, the kinase, that one. Enzyme that catalyzes transfer of phosphate groups. High energy to something else. High energy to specific substrates. And then this was for this fission yeast. Yeah, okay. I like to keep that one open, I think. Major histocompatibility complex. The dimer is the oligomer consisting of two monomers joined by bonds that can either be strong or weak. Okay, cool. So what does this do? Chief currently serves on the board of directors. He received this. Okay. Is there some engineers here? Intellectual property. Ah, they don't put their real workers in, do they? Heads and leads. Let's get these names down. Samarth Kulkarni is the CEO. Philippe Drouet, CCO. Dr. Tony Ho, Research and Development. Lawrence Klein. Samarthi Kulkarni, Dr. Tony Ho, Philippe Drouet. Let's have a look what these guys say. Samarth Kulkarni. How do you spell his name again? Samarth Kulkarni. Samarth Kulkarni. CEO at CRISPR. Oh, is he a new guy? Cool, new guy, alright. We like new guys. Right, I need to follow this guy. People. Contacts. Right, be right back. I'm gonna just take a quick break whilst I do a blood test. So, one secarino. I'll mute myself as well so I don't annoy people. Alright, cool. Um, so we've had a good look at CRISPR. I think I know what it roughly does now. So let's go back to this plan. How do you break up a DNA sequence CRISPR? We've got loads of detail on that to read through. And I think I could just pretty much come back to this site. I don't have a worry about reading offline about that. So how do you put the DNA sequence back together? This is still using CRISPR. I'm glad the technology exists to put it back in. So CRISPR actually, I mean, let's just get the proof of that again. My brain is not great. So it says delete and insert, yeah. So that that's awesome. So one technology will do it. Here's the question: are there any other technologies? If so, what do they do different from CRISPR? Why have I only heard of CRISPR? Um, Yeah. What's the most popular? Like, if I imagine like thinking about coding languages, which ones you choose because they're better, or open source or all that stuff. I want to know all my options basically. And what's the price planning of this? What do I have? Is this like a licensing thing or is it just open source stuff? Partnerships. A core component of our strategy allowing us to access capabilities and resources to support our therapeutic programs. Huh. With an email. Please contact us at this email. Interesting. BD. Becton Dickinson. What? No, can't be. No. I know these guys. Okay, it doesn't really say much, does it? Patience. Okay, so we know something about the body using symmetry in DNA. If we found that out, so that when one side of this DNA is corrupt, then it will hunt out a replacement. This is pretty much what this was saying. Um, with this, what was what's the word for it? The terminology. Saw it here somewhere. Oh no, wait, it was in that video. That's right. It was in the video, or maybe it was this one. DNA-associated virus. Is corrupt, then it will hunt out a replacement. No, adeno-associated. No, this is the virus thing for hunting. Guide RNA. That tells it where to put itself. Yeah. Okay. So learn about what the symbols mean in chemistry and biology. Okay, that's a big topic. Good source of white papers. Yep. Okay. Let's just focus on. Let's see the priority here because I think this has to come as like a side note. Let's have a look at these machines. I think. Let's look at these machines. Okay. So, pipetting machines. In fact, let's see if there's any YouTube videos of like CRISPR experiments or something like that. I mean, I did search. Enter doesn't work. That's interesting. Let's try that again. I'm using Firefox because I didn't want all my URLs to pop up when I was searching. Meet the biohacker using CRISPR to teach everyone gene editing. Yeah, yeah. Oh, dude, this is the dude. Oh, yo, that's my boy. Wait, this looks good. The CRISPR system originated in bacteria. It is more commonly used to edit eukaryotic genomes rather than bacteria. Let's have a look at this. What this one says.
Right. Who's eating all the quavers?
I think I have to mute these, don't I? I don't know. Am I allowed to see if we can get some subtitles going? Ah, go away ads. Let's see. Uh, I just want to see what kind of bacteria. One gene editing method is gaining a lot of traction is a CRISPR-Cas9 system. Bacteria is blah, blah, blah. Ah, because most bacteria do not possess robust DNA repair systems. Double-stranded DNA breaks are typically lethal to bacteria. Okay, okay, okay. Aha. Look. Yes, let's do this. Yeah, teach me this. I think I have to put this on. I can't actually.
Cassette in E. coli. This case study will cover 3 phases. Phase 1, the design and cloning of Cas9 and sgRNA for bacterial
Ah,
systems.
talk is so slow.
Phase 2, preparation Preparation of electrocompetent cells and transformation. Phase 3, screening and confirmation of gene knockout. In phase 1, 3 components are prepared for CRISPR gene editing in E. coli. 1. A plasmid with the sgRNA. 2. A plasmid carrying Cas9 and the lambda red genes. And 3. The repair template. First, sgRNAs are required to guide the Cas9 nuclease to the target locus. An sgRNA was designed against a chloramphenicol.
sgRNA, something guided. I didn't even see what that said now. Ah, damn it. I was just too thinking. I was thinking too much about how slow it was. Okay. That's interesting.
Acetyltransferase cassette, or CAT gene, which was previously introduced into the E. coli genome. When intact, this gene gives resistance to the antibiotic chloramphenicol. The sgRNA was cloned into the P-target plasmid. Second, a construct carrying the lambda red genes is required. The lambda red genes encode phage-derived proteins that are crucial for enabling recombination of DNA fragments. The PCAS plasmid carries inducible lambda red genes, as well as constitutively expressed Cas9 and a kanamycin resistance gene. Finally,
Kanamycin. Antibiotic used to treat severe bacterial infections and tuberculosis.
facilitate recombination, a repair template is required. This template contains left and right homology arms that are
That's what I want. I don't care about this.
Writing's not that easy.
Mute. Alright, this looks good.
Is CRISPR-Cas9, and this is really revolutionized molecular biology itself. In this video, we'll look at a little bit about the use of the CRISPR and how CRISPR can be achieved in the laboratory. So CRISPR-Cas9 systems can be used to generate knock-in, point mutations, and knockout lines. So all sort of genetic manipulation at DNA level is possible using CRISPR. Not only...
What is this? So genomic DNA, Cas9 kind of encompasses this part. It's interesting how they show the diagram going over that. RNA, trace RNA, PAM site, and they can cut certain positions. They got knock-in, knockout, point mutation, genetic manipulation, imaging, genomic loci, CRISPR fish.
DNA. CRISPR has a brilliant way of targeting a genomic locus. So a particular genomic loci can be highlighted and can be imaged using CRISPR-mediated fish. If you add fluorophore with the Cas9 enzyme and allow the guide RNA to target a particular genomic loci, so the genomic loci would be highlighted, and that too in a very specific manner. Then, at RNA level, or at RNA production level, you can also control. You can perform transcriptional activation and transcriptional repression paradigm using CRISPR. So in this paradigm, you use the guide RNA to target this machinery, and this particular Cas9, instead of cleaving, just stay there near the promoter. Now, depending upon which sequences they are binding, they either allow or facilitate the binding of the RNA polymerase, or they might hinder the binding of RNA polymerase. As a result, transcriptional activation or repression could be achieved. Not only that, the futuristic gene therapy could be...
This is interesting. I can't quite see the... YouTube. Let me just try and zoom in on that. Okay. I don't quite... so transcription activation is saying that this CRISPR-9 is cutting one... I think this might be that cut one side off thing. It's yeah, because I remember the video now. It was saying that it kind of unwinds the sequence. I'd like to know more about it. And I can't remember what this is. Is this the... Okay, fine. Dunno.
Or the custom-built medicines could be designed using this CRISPR strategy. Yet we are far away from it, but it's not so far. There are possibilities. Now let's talk about that how CRISPR can be achieved using cell culture method, or how it is performed in the laboratory, how the scientists can perform it in the lab.
Dude, calling out animated biology with ARPAN. Heck yeah, dude. This is exactly what I need.
So it is possible now to manipulate human cells, human stem cells, using CRISPR-Cas9 technology. In this video, we are going to look at that, so stay tuned till the end of this video. Exactly, it would give you a flavor how things are done in the lab. So here are some cells, which are human embryonic stem cell lines, in a plate. Now what we are going to perform is to perform several genetic manipulations in these stem cells. Definitely the gene that we want to manipulate, we are going to use a guide RNA against it, and the CRISPR machinery would cut it. And inside the cell, there would be repair machinery such as NHEJ or homologous directed repair mechanism. And when this repair mechanism would happen, we would incorporate our changes. So in order to perform that, we have to add a mixture of the cells, several plasmids, about which I'm going to talk about, and a plasmid which would help you to screen.
Right, this is it. This is exactly what I wanted. Oh, thank goodness, dude. Thank goodness. Right. CRISPR. So it looks like you you can actually, yeah, petri dish looks absolutely doable. Inject plasmid. I didn't quite catch what it was saying, so I'm gonna just go back a bit.
Mixture of the cells, several plasmids.
Whoa, 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 whoa.
Manipulate. We are going to use a guide RNA or homologous directed repair mechanism. And when this repair mechanism would happen, we would incorporate our changes. So in order to perform that, we have to add a mixture of the cells, several plasmids, about which I'm going to talk about, and a plasmid which would help you to screen. In this case, it's a transient GFP expression plasmid. It would only tell you that your transfection reaction has worked properly. The next step would be electroporation. So electroporation is one of the efficient way of transferring gene inside the cells using electric current to form small
Okay.
pores on the cell, and the DNA, that means in this case the plasmids, would get inside the cell. After that,
Okay. Okay. So you inject this plasmid varieties. You then apply electrocurrent. I think they call it electrotransport. Allows the plasmid into the what was it?
pores on the cell, and the DNA, that means in this case the plasmids, would get inside the cell.
Inside the cell. Okay. Inside the cell.
After that, you take the transfected cells or electroporated cells and you plate them on a fresh plate and allow them to grow for a little while.
Allow growth.
Now, since we have expression
allow growth of end result of the above. Alright, this is good information. Oh, we're gonna hack the freaking genome, dude.
plasmid for GFP, so the transfected colonies would be highlighted in green if you see under a microscope. Now, there is a chance that the colonies which would be green or which would be GFP positive, they also have other plasmids which is required for the CRISPR. So most probably, some of these colonies would be positive for this genetic manipulation. So in the next step would be sorting out all the GFP positive cells from these transfected cell pools.
Fact sorting. Nice.
After the fact sorting, when you really sort GFP positive cells and collect them separately.
Let me just see what machine that was again. Fact sorting. I won't forget that. Come on, YouTube, don't fail me now. Come on. Right. Fact sorting. So you got some kind of computer running the whole thing. What the hell is that? What is that? It's like some printer? I think this is... okay, so this is the Petri dish thing. Is this inserting them into this kind of chamber of things? I guess it's... just total guessing, but is this the electronic thing? God. Fact sorting. Was this all to do with fact sorting? Okay, that makes more sense. Okay.
These transfected cell pool. After the fact sorting, when you really sort GFP positive cells and collect them separately, you put those GFP positive cells in a new plate and allow the cells to grow for a while. After the cells grow, you would see in the plate there are colonies, and these colonies are achieved from one single cell which was GFP positive. That means which was
Fact sorting. I want to know what that is right now. What is fact sorting? Fluorescence activated cell sorting. Specialized type of flow cytometry. It provides a method for sorting heterogeneous mixture of biological cells into 2 or more containers, one cell at a time, based upon the specific light scattering and fluorescent characteristics of each cell. Ooh, nice. Okay.
Transfected. At least we know the transfection has worked on those cells. But we yet don't know all the plasmids are present in all of these colonies or not. In one colony, it is possible that all the 3 plasmids are present. In one colony, it is also possible, let's say 2 plasmids are present, the third one is not there. So what we really don't know in which colonies all of the plasmids are present and all the genetic manipulation has taken place. So what we have to do under the microscope, we have to put collagenase and take out all of these colonies and distribute these single colonies into 96 well plates or a 24 well plate.
Yes. Okay. Yes, this is exactly what I wanted.
Each well, you put 1 colony, and this 1 colony is derived from 1 single clone. From this, you do something which is very similar to replica plating. When this plate is confluent, you split them into 2 different plates such that one plate is exact replica of the other. From the material present in one plate, you extract the DNA and use PCR-based screening or sequencing-based screening. And
PCR-based screening. That's... PCR is... Shit. PCR. What does PCR stand for? Polymerase chain reaction. Polymerase chain reaction. That is definitely a key word. We've got to get that down. CRISPR. Polymerase. Wow. Polymerase chain reaction. Oh yeah. Okay. Get in my brain.
let's say after screening, you figure out that these 3 wells are actually positive for whatever you want, whatever genetic manipulation you wanted to do. So you track back.
So there's a failure rate of this. Oh. Oh no. Okay. Why is there a failure rate on these things? Why is there a failure rate? New questions. Why does the process of growing plasmid injected electro-translated copies have such a high failure rate? Seemingly from the video. Do you want to not? Didn't mean to do that. Why does the process of growing plasmid injected electro-translated whatever it was copies have such a high fail rate? At least from how it seems. So, yeah, in the video. Okay. Oh, that's really annoying. So I wonder what the waste is on this. Yeah.
To the root plate and take out the cells from those plates and expand them and keep them for future uses.
Let me just see if I understood this correctly. I want to make sure I understand this. So you inject the result of the colonies into these different things. You split them up. You then separate them. What was
From
this?
this, you do something which is very similar to replica plating. When this plate is confluent, you split them into 2 different plates.
Confluent. Yeah, I don't know these words. Sorry. Define confluent. Flowing together or merging. When these things are confluent. I'm not sure that really explains it.
You put 1 colony, and this 1 colony is derived from 1 single clone. From this, you do something which is very similar to replica plating. When this plate is confluent, you split them into 2 different plates such that one plate is exact replica of the other. From the material present in one plate, you extract the DNA and use PCR-based screening or sequencing-based screening. And let's say after screening, you figure out that these 3 wells are actually positive for whatever you want, whatever genetic manipulation you wanted to do. So you track back to the root plate.
So this is going to have to do this pipetting thing that I was talking about to find out which ones are yeah, positive colony, as they say. So I'm gonna need such a huge amount of this to be able to do AI-based tests. Like, can you imagine if I do experiments I don't even know what's happening? I'm gonna have to know up front. I'm gonna have to know up front what I want to do, because the testing would be massive. You know, if I so let's say I want to go and cure diabetes, I run AI on it. AI might take, I don't know, God forbid, like trillions of iterations to try and figure out anything useful. That's all of these need to go through testing, and all of these might be wasteful. So I wonder what the maths is for waste over like actual sequencing tried. Ah, it's so annoying.
And take out the cells from those plates and expand them and keep them for future uses. And this is your cell line. You revalidate with sequencing as well. So let's look at what are the plasmids. Here, the plasmids.
Oh, hang on. Unless he means, is it just that it hasn't grown yet or something, or hasn't done its thing yet?
So you track back to the root plate and take out the cells from those plates and expand them and keep them for future uses. And
Keep them for future uses. Cell line needs to be expanded.
This is your cell line. You revalidate with sequencing as well. So let's look at what are the plasmids. Here, the plasmids are depending upon what type of manipulation we are doing. In the first case that I'm going to share with you, we're going to do a knock-in experiment. That means we're adding some component inside the genomic DNA which was not present. Many of the cases, scientists attach a GFP or a fluorophore coding gene to mark many proteins endogenously. In this case, the donor plasmid has that sequence. Another plasmid has the sequence for Cas9 and also had the guide RNA, which is shown here in the black. The third plasmid is a transient GFP expressing plasmid, which would tell us the transfection has worked or not. So the guide RNA would target the Cas9 system to a particular genomic locus, which it should be targeted. And then the donor plasmid have sequence homology with the strand which is targeted. And based on homology, it would be actually incorporated in the place of...
That's amazing. Like, once again, the same process, which this homology will mean that it tries to take it on board. Donor plasmid. Injecting something that you've modified, I hope. Or corrected, yeah. See, oh no. Does it mean that you can only correct what was already there, rather than insert more computation? Oh no. Okay, let's hope I'm wrong. Let's go, I know, 1. I don't think this really works very well with images. Let me see. Whoa! Can I? No. Oh my god, what am I doing? No, stop. How do I go to the next bloody line? Just delete this. Is that the YouTube video? That's the YouTube video. Who cares then? Cool. So, does CRISPR work for gene therapy rather than full manipulation of everything between the two join points? I wonder if the homology of this will let me just make sure I'm using the right word. Homology. So having the same or yeah. I wonder if the homology of this will mean that the DNA won't take it back in if it's like too different. If that's the case, this really puts a stop to anything I want to do. I hope I'm wrong.
The double strand break, and a homology dependent repair would take place using this strand, the donor strand as a template. As a result, the donor strand should be incorporated into the genome. And using specific PCR primers, you can amplify the particular region, because you know this particular GFP containing region is not present in this cell, in the cells normally, right? So the wild type would not show this band, but the mutant or the knock-in line would show a band which would amplify a portion of the GFP and the portion of the normal genomic DNA. So using that screening method, you can have a rough idea which cell lines are positive. Then definitely you would send it for sequencing to understand the incorporation of a sequence. Later on, you can also
send it to sequencing. How do we sequence something? Let's have a look. Machines for sequencing DNA. Illumina makes a number of next-generation sequencing machines using this technology, including the high-sec genome analyzer IX, blah blah blah. Process microarrays. Wow. Technology leading to these DNA sequences was first released by Select 2006. Let's hope they've upgraded it. A scientific instrument used to automate blah blah blah, giving us used to determine the order of the 4 bases. They've actually got names. I never knew that. Ha, makes sense. Process text strings. Yes, how do they do it though? Actually, this is essentially what I need to do. Like, I need to study stuff I already know before even being able to attempt to CRISPR stuff. Can also use optical instruments as they analyze light signals originated from fluorochromes attached to nucleotides. Ah, okay. Okay. Some DNA sequences can also be considered optical instruments. Okay, I wonder what there's like the kind of fail rate of these ones are, considering it says some. I don't care what the first one. Tell me on this now. Reads are short. More recent third generation DNA sequences such as SMRT. Oxford nanopore. Yes. Get in my brain. Search keywords. Let's call this DNA sequencing. At what point does someone think that when I copy something I want any of this, and they give me the option to remove it? Why is it not default the other one? Fuck's sake. So stupid. Okay. More recent third generation sequences such as SMRT. Okay, let's have a look at these. A single molecule real-time sequencing. Parallelized single molecule DNA sequencing method utilizes a zero mode waveguide, a single DNA zero mode waveguide. A zero mode waveguide. Nope. It's an optical waveguide that guides light energy into a volume that is small in all dimensions. So this is that optical thing it was talking about. Cool. And what was the other one? Oxford nanopore. Yeah. Is this? Okay. Used in the 60s by. Yeah, because I think if this Oxford is what I think it is, then this is the thing that's kind of setting UK above others in terms of like DNA sequencing. I wonder if it's what they talk about. That a need for PCR amplification. So okay, this is good. Using nanopore sequencing, a single molecule of DNA or RNA, a single molecule, can be sequenced without the need for PCR amplification or chemical labeling of the sample. Chemical labeling of the sample. At least one of these aforementioned steps is necessary in the procedure of any previously developed sequencing approach. Nanopore sequencing has the potential to offer relatively low-cost genotyping. Ooh, sounds good. High mobility for testing and rapid processing of samples with the ability to display results in real time. Publications on the method outline its use in rapid identification of viral pathogens. Yeah. Monitoring Ebola, environmental monitoring, food safety monitoring, human genomes. Okay. Yeah. Okay, let's get this. Nanopore sequencing, we've got it. This seems really important. Okay. So if I go back to this video, send it off to a DNA sequencing. So that's what he said before. So let's see what.
Sequencing to understand the incorporation of a sequence.
Mm-hmm.
Later on, you can also use the same principle to target the guide RNA, and instead of having a donor plasmid, you put a repair oligo. And this repair oligo is basically having a point mutation inside of it. Now, when this repair oligo is replacing in the double-strand break, it incorporates that mutation inside the genome. And you have several ways to understand it. Many of the cases, a new restriction site would appear there, which was not previously there. So you can cut it with a different restriction enzyme. It would give you a particular restriction bands. But the ultimate way to understand the point mutation is to perform sequencing. That would tell you exactly the sequence change has happened or not. Though
we're looking at here. I wonder what this is. Because you have this these peaks. I wonder if they're quantity. Obviously, it can't be. It can't be. It can't be. It can't be quantity. There's something very specific I need to learn here. It's got this like subgraph going on here too. Oh no, no, it's not. I see. I see. It's just overlapping. Okay, I got you. It's funny how it kind of peaks a little bit. This is like the binding, the kind of relationship between the two. I wonder if this is when it tries to understand what it's looking at. So it's like, hmm, oh, I know, I see. This is how much light is coming from it. So each one is going to show a little bit more than it should for everything. But the main point is that this is the most amplified amount of light coming from it, or color. PCR, the oh shit, I've taken it off now. Oh well.
CRISPR looks highly precise, high throughput, but you need to screen as many as hundreds of colonies to get specific mutants. So this is also a tedious process as well in the laboratory. But
yeah, that's what I want to get rid of, dude. We want to get rid of tediousness.
This process has a lot of promise and it is really, really targeted. So with that, I would say goodbye. And if you like this video, give it a quick thumbs up. Don't forget to like.
I will give it a thumbs up, don't you worry. I'm just not logged in here. Wow, that was awesome. That was absolutely awesome. I feel like watching it again. How's everyone doing? No one's doing. Okay, that's great. Cool, just me learning. Always good. Let's see what's happening here. Cool. Okay, so that was awesome. That kind of proves to me that I can actually buy some of the kit, because I can do I can get the dishes, I can get a machine that does that kind of automation for me. I need to understand the APIs that are available to use, and that's one of my deep dive things as well. This is a deep dive for like loads of sessions. This is just the kind of first entry into it all. Yeah, so what kind of machines kind of started to answer that already. Simulation tooling, is there a day specific cool words that instead are named in a functional form? Or you know, it's funny, this is actually really good, but I don't think it's what I want. The gene cards. I have a funny feeling this is going to be super helpful though. Yeah. And then symbols of chemistry biology, it's an ongoing thing. Good source of white papers regarding engineering of DNA sequencing and manipulation hijacking. Yeah, I think I should probably keep looking at machines then. Let's see how much these things, all these bad boys cost. So first things first, I need a pipetting machine. So I can put the plasmids into the solution, the DNA mix, the cells. That's right. To grab the DNA from the cells. That makes sense. So pipette max, automated pipetting robot. Ads. Opentrons. Ah, look, to a lab with the next gen $5,000. I can afford that. We can do that. We can do that. Lab automation has never been easier. Nice, good. Looks good. Let's see what we've got here. Wait. This literally says from 5K. Oh, don't you worry, my friend. We will be coming back to you. The high precision OT2 lab robot is flexible open source. Open source! Whoa. Ah, so many things. A GitHub. Oh, what? Yes. Software for writing protocols and running them on Opentronics. OpenTrons. Yeah, baby. What have we written in? Let's read me. Makes famous biologists find this has a common platform to easily share protocol and blah 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 blah. Simple framework designed to make writing automated biology lab protocols easy. Yes, come on, dude! I can't believe we just found this. We designed it in a way we hope is accessible to anyone with basic computer and wet lab skills. Oh yes! As a bench scientist, you should be able to code your automated protocols in a way that reads like a lab notebook. Okay. Pipette.aspirate. Pipette.dispense. Location. Table of your a table of your dishes. How much you're gonna put in. Dispense it on the well plate. Oh my goodness. Wait. Python. It's written in Python. Oh my god. Let's keep reading. I'm getting too excited. The OpenTrons API is a simple framework. Blah, blah, blah. This example tells the OpenTrons OT2 to pipette 30 microliters of liquid from a trough to well plate. Learn more here. Documentation, source code. Let's have a look. Okay. Yeah. The design goal of this API is to make code readable and easy to understand. For example, below is a short set of OpenTrons. Oh, dude, I will not forget your name. Let's just get this out of my head and into a note. What we found out. What's with your URL? Disgusting. Provides API-driven pipetting machinery. Fuck, is it E? It's an E. Of course it's an E. Yeah. Read blah blah blah. From well A1 to well B1 that even a computer could understand. This protocol is by me. It's called OpenTrons Protocol Tutorial and is used for demonstrating the OT2 Python Protocol API. It uses version 2 of this API. Begin the protocol. Add a 96 well plate and place it in slot 2 of the robot deck. Add a 300 micro tip rack and place it in slot 1 of the robot deck. Add a single channel 300 pipette to the left mount and tell it to use that tip rack. Transfer when okay. If we were to write this with the Python Protocol API, it would look something like the following. Protocol name, blah blah blah, okay. Define run, where's the run? Run has not been here, okay, that's from wherever. It takes a protocol context. And so what we're doing, so adds a 96 well plate, so load labware, Corning 96 well plate, 260 microliters flat. I wonder what this is, but that's cool. 2 in slot 2, okay. Tip rack. Place it in. Right, okay, let's get these names. Well play. Oh my god, this is amazing. Well play, of course it is. Let's get that in my search keywords. No sequencing, CRISPR, pipetting machine. Well play. Yeah, we know what that is. Let's get a tip rack. What's a tip rack? Oh, I see. This goes into these. Okay, nice. Tip rack, tip rack. Left pipette. Load instrument. 300 singles. So this is a single channel 300 micro pipette to the left mount. Left pipette. Load instrument. Load labware. So load the actual hardware, then actually put the instrument in. Add a single channel, perpetual left mount, and tell it to use that tip rack. Load lab where load instrument 200 single to the left. Tip rack is here. Gonna put it into there. Oh, that's so easy. It's so easy. It's cool that it's an array as well, so you could do multiple things at once. That makes sense. Pick up tip. Transfer 100 from the plate's A1 well to its B2. Pick up tip. Aspirate it. Dispense it. Drop tip. That's exactly what I need. Opentrons. So what, let's see. Start guide, product help, install the app. I cannot believe that. And GitHub, I wonder if it's, oh, they've got a little app as well. Oh no, is it Mac? No, it's Python. Yeah, yeah, okay. Hmm. Okay. This looks good. This looks very good. From 5K, so schedule demo. Order online, let's have a look. Ooh, from 2. I wonder what the 2 part is for. P20 single channel Gen 2. What does that actually mean? So this is the OT2. And it says left pipette is a P20 single channel generation 2. P20. What does that mean? P20. Oh, give me a bone, please. Give me a bone. Ah, nothing. P20. Pipette. Okay. I see. So it's actually like the utilities that are used for the machine. It's like a standard. Whoa, shit, that's expensive. Okay. How many does this run? Oh, please don't tell me that's per... No, okay. So I see. So it goes into this machine here. Yes. I bet that's what it is. And this kind of automates itself like kind of thing. Okay. I think I understand that. So what's the difference between 300 and 1,000? I guess it's like quantity that it can inject. Classic P20. Where's like the amount? Yeah, volumes from, okay. Is it nanoliters? I can't remember. Well, let's have a look. Microliter, gotcha. So 0 0.2 microliters to 10 milliliters. 10 milliliters. I wonder. So I need to ask another question. How what size of plasmid injection is needed for DNA sequencing? This will decide the size of pipetting tool that I will need to purchase. Okay. Oh, add to cart. That is just so insanely crazy. God, I'm tired. Okay. So yeah, we're looking at what the difference is. P300. Let's have a look. P300 pipetting. Okay. Oh, Gilson again. These guys are going to come up a lot. Unexpected. Don't sell them anymore, maybe? Single channel Gen 2. You can get it without them. P300 8 channel. P300 8 channel. Yeah, it's just bigger. It's gone from what was it, 0.1 a 0.2 to 10 mils. Ah, here we go. Ha, yes. It just means bigger. Okay, gotcha. Understood, understood. Understood. Okay. Left pipette takes from one tray and then inserts into the other. That makes sense. Oh, cool. There's a video as well. Let's have a look.
Hi, I'm Taylor Goodman, Director of Hardware at OpenTrons.
Good man.
I'm going to explain some of the hardware design decisions we made in the OT2 to create the most affordable, high-precision liquid handling robot ever sold. First, let's take a look at the overall gantry system. Our goal was to create a rigid and precise structure using affordable manufacturing techniques. The main structural components, including the gantry, the frame, and the bridge, are high-precision extruded and CNC'd aluminum, creating a rigid design that can be built at high scale and at a low price.
This is essentially a biological 3D printer. This is so cool. But it does have the process of taking, yeah, because the 3D printer takes materials from a place and then puts them down, right? Or does it just have everything inside its arm? I don't know. I need to probably do some 3D printing.
Structural integrity comes from the two sheet metal side panels on either side of the machine. The precision aluminum deck is machined to a high tolerance and is removable to allow for additional configurations in the future. The robot is
yeah, and to allow James to clean it because he probably spills everything
enclosed
everywhere.
in clear acrylic and comes with covers for the vents in the floor if you'd like full enclosure. The door has a friction hinge to stay open when you are loading and unloading the deck, but for safety, the robot will not run if the door is open.
This isn't enough though. This is tiny. This is too small. But I can do some beginning experiments. So I'm thinking, I've already said it before, but if I need to do AI driven analysis of sequencing, number one, I need to do the sequencing on a massive scale, and number two, I need to be able to pipette these plasmid things in, and I'm essentially just going to be doing lots of tests. So if I'm going to do lots of tests, that means I've got to be really specific about what I want to do. That means I've got to know what I'm doing. Okay. Because, like, I assume... Well, maybe there's different sizing. Anyway.
Next, I want to tell you about our new robotic pipettes. The pipettes are the business end of the machine, and we took a lot of care to ensure accuracy and precision are comparable to machines 10 times more expensive than the OT2. You can easily install them into the machine with an Allen wrench we've included with the robot. The OT2 can hold 2 pipettes, single channel or 8 channel. It doesn't matter which side of the head the pipettes are mounted to, and you can use 2 multi-channels or 2 single channels at the same time if you prefer. Here's a look inside the pipette. We use a robust microstepping motor attached to a precision molded volume chamber. The volume chamber is a cylinder with a piston that converts the linear motion of the motor into the desired volumetric output of the tip attached below. The tip cones are made from a hard composite plastic designed to put on and eject hundreds of thousands of tips throughout its lifetime. You can learn a lot more about our pipettes, including the specific plastic designed to put on and eject hundreds of thousands of tips throughout its lifetime. You can learn a lot more about our pipettes, including the specific linear millimeter to microliter calculations for each volume range on our white papers found on the pipettes page.
The calculated volumes from both gravimetric and photometric testing were then averaged and compared with intended volumes. We assessed random error or precision using the following equation. CV is the coefficient of variance. Omega is the standard deviation of the sample set. Wait, what is that? It doesn't really matter. X is the mean of all the sample volumes. CV is the coefficient of variance. It's a standard deviation. X is the mean of all the sample volumes. Yeah, okay. We assessed systematic error or accuracy using the following equation. X minus test. Why are they telling me this? I guess science. Like, can't they just give me a percentage of?
Okay, that's all for this video.
Can you tell me how the failure is? What's your failure rate, bro? Accurate, dispense 1 microliter into 384. Flexible, fast, user-friendly, open, compact. Where's accuracy of like? Oh no, why did you mention fail? Because now I want to know what you fail on. This looks good. I could do some calculations for that. 96 oil plate in 22 seconds. Slow though. It's gonna have to be really, really specific about what I need to do. There's a lot of learning here, jeez. Yeah, let's watch the rest of this video.
Each volume range on our white papers found in the pipe to better understand our new lab robot. If you have any more questions about the OT2 hardware, you can look at our documentation or chat with one of us online. Thanks so much for watching.
Awesome. Love it, mate. Love it, mate. Brilliant job. Get a free one-on-one demo as well. Look at that. Heck yeah. Alright. Wow, that's kind of answered loads of questions. Jeez, and that's such a good price as well. What is that in? It's not a lot. Oh, you are absolute muffin face. Dollars in GBP. Yeah. It's just a little bit more than the computer. A little bit more than the computer. And then you've got to buy this. I wonder why you'd need to buy replacements. Legendary accuracy. Gilson. Great Britain. Interesting. Not so Great Britain. Compact footprint. Single and multi. Oh yeah, how big is it as well? 63 by 57 by 66. Oh, it's fine. Okay. Video and audio, integrated video camera, and audio system included with the feature all custom. Oh, that's interesting. I wonder if I could do. I wonder if I could do something with that camera to help out as well. adding cameras in there to kind of study what's happening. I doubt they'll be efficient enough. Cool, I like it. So what else have we got to do? What kind of machines? Prices. We've got a rough price on one of them. Automated pipetting. Yep. Logistics around supplying with more materials of the process. So this is like, so what are the actual chemicals and stuff we can put in these things? This is all glass, right? So let's investigate what plasmid is. So chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. They are most commonly found as a small circular double-stranded DNA molecules in bacteria. However, plasmids are sometimes present in archaea and eukaryotic organisms. Plasmid for purchase. Plasmid solution. Centrifugation. Oh man, is this going to be another step I've got to do? Can't you just buy vials of this stuff? So so let's imagine I want to just sequence my DNA. So I need to get some of my cells. I plop them in a dish. I inject the necessary. Oh no, hang on. Yeah, I do the micro, no, the nano, the thing that the Oxford guys did. What was that? The fax? No, it wasn't that. Ah, but it was the other one. It was the nanopore sequencing. Okay. Okay. I wonder how much time I've got left. How many hours I've been doing this now. I'm feeling a little tired, but yeah, just over 2 hours. Okay. So I think last thing I'm gonna do tonight is gonna just see some white papers on DNA sequencing. I know that Manellus Kellis always fires his stuff out. He's just an absolute legend at MIT. It's on DNA sequencing, let's say on CRISPR, I guess actually. Latest papers on CRISPR discoveries. CRISPR-Cas9 genome editing exploits the CRISPR-Cas system to modify genome editing. We know that. It's all good. Latest research and reviews. Accelerating target deconvolution for therapeutic antibody candidates using highly parallelized genome editing. CRE-controlled CRISPR mutagenesis provides fast and easy conditional gene inactivation in zebrafish. CRE-controlled CRISPR mutagenesis. Mutagenesis? What's muta mean? Genesis. Process by which the genetic information of an organism is changed by the reduction of mutation. It may occur spontaneously in nature as a result of exposure to mutagens. Got you. Simple and reliable detection of CRISPR-induced on-target effects by something guided PCR. Can't remember what PCR is again. Do I have it here? No, I do not. Prime design software for rapid and simplified design of prime editing guide RNAs. This sounds good. Prime editing PEs, versatile genome editing technology, but design of the required guide RNAs is more complex than for standard. So okay, let me just read this. Here we describe Prime Design, a user-friendly end-to-end web application and command line tool for the design of PE experiments. Prime Design could be used for single and combination editing applications as well as genome-wide and saturation mutagenesis screens. Using Prime Design, we construct PrimeVar, a comprehensive and searchable database that includes candidate prime editing guide RNA and nicking sgRNA combinations for installing or correcting. What? Who uses that word? Or correcting 68,500 pathogenic human genetic variants from the ClinVar database. Finally, we use Prime Design to install a variety of human pathogenic variants in human cells. Okay, cool. Oh, this is perfect. I didn't know nature.com did this stuff. Okay, helpful pages links. So you got the awesome gene card informational database. Informational database. Then we've got this white paper site like that. Let's see if I can find anything more. I think maybe the medical database. MD, where are you? Publications, here we go. This isn't the one I meant, but PubMed's good. DNA, CRISPR actually. Let's do CRISPR. Ooh, nice. CRISPR. CRISPR-Cas9 for genome editing progress, calculations and challenges. Clustered regularly interspaced short palindromic repeats. CRISPR-associated protein 9 system provides a robust and multiplexable genome editing tool. Oh, this is interesting. This is really interesting. These guys, this is twice now I've had this like tool thing that they're given rather than studies. Precisely manipulate specific genomic elements and facilitating the elucidation of target gene function in biology and diseases. CRISPR-Cas9 comprises of a non-specific Cas9 nuclease and a set of programmable sequence-specific CRISPR RNA, which can guide Cas9 to cleave DNA and generate double-strand. What time is this? What date? Damn, that's quite old. Yeah, this makes... I wonder how old the other study was. This is new. So can I sort this by date? Publication date, last 5 years. Oh, we've got it. This is it. I've got to go through and read these. Okay. Let's do that. And let's get one more. One from Manellus Callus. Nature.com. Ha! Oh, this is... That's great. That's exactly what we need. I can't help but think I've just streamed this with a massive screen. Blatantly have. Let me just double check. We live and learn. Output. Video. Oh no, cool. 90 by 1080. Glad to hear it. check this out. So, where's the references? Where are they? Guess they don't have any. Okay, these are in reference to. Okay, uniform processing, signals, biosamples, enhancer biosamples, DHS index, imputation, validation. 1. Uniform processing, imputation. What is that? I can guess. Charge or claim that someone has done something undesirable. Okay. Is there a biological thing? Biology. Let's try that. Genetics refers to the statistical inference of unobserved genotypes. It is achieved by using known haplotypes in a population. For instance, from the hap map or the G. Oh my God! Look at this. Hap map. Let me just add that to my search keywords. That will be. Let's call this. Genetics. Hap map. Okay. Where are we? Here. 133 biological samples. 2.1 million enhancer locations. Links downstream genes. Motif issue enrichment upstream TFs. I wonder if any of this really, like, it's more of a kind of feel of the data, isn't it? It's not like very specific. But here it's saying, well, let's look at what we got. Diabetes. If we're going to maybe pancreas. Pancreas. Here we go. So this blue. Oh, this is quite common. Yeah. Oh, let's try something a bit more obvious. This yellow here. No, maybe the pancreas is fine. I can't tell what the colours are. It wouldn't help to be colourblind right now, I don't think. Sheesh. I can't tell if that... Ah, it's these ones. Okay. So what is this saying? So we created a compendium of over 17,000 epigenomic tracks across 18 marks by uniform processing and imputation and used these to call chromatin states for 833 bars of and active enhancer states over 2.1 million DNAs. Hypersynthesis sites. We used unsupervised clusters of the enhancer activities to call enhancer downstream target genes, upstream regulation, and to prioritize, investigate, and compare hundreds of GWAS traits and thousands of loci. GWAS. Genome-wide association. Gotcha. Gene regulatory studies, complex trait investigation, studies of disease locus. This is great. This is exactly what I need. This kind of thing. So this is nature.com. So Manolis Kellis is actually quite good here. Helpful links. Manolis Kellis provides good references to MIT specific research. This was MIT, wasn't it? Research. Yeah. Okay. Well, it might be for his own company or something. I think they do research in the MIT labs, though. So we got. Oh wow! Look at this data. So I think that they're kind of onto this stuff already. Looking from this. Nearest observed data set. Look. Average observed data set. What is this? Can I see into here? Can I zoom in? Oh yeah, we can. Wow, it's so slow. Oh my god, yes, okay. That's my 2 hours. Stop. Cool. Well, I'm feeling pretty tired, so I think that's a good like little insight, little start into what the hell I'm going to be looking at. I've just got to start warming up a bit with it all. It's a little bit too much at the moment. So thank you for watching if you watched, and if you didn't, you probably didn't miss out on much. I'm gonna try and do 2 of these deep dives every week. So might be something like Wednesday and Fridays. I guess it depends on how the weather is and how COVID-19 goes. Um, this is a massively ongoing thing. So for the next, I don't know, 10 years or whatever, I just want to start dipping my toes in slowly, um, learn what's out there, and then I'll start to kind of leak out some of the actual problems that I want to solve. But mostly this is just I need to kind of get an understanding and feel for everything in the genome sequencing, DNA editing world, and the kind of machinery that I might be able to buy to do these experiments myself. I might also do a deep dive on AI. I've had a play with AI already. It's pretty math heavy, but don't worry, I got a T-shirt of mathematics, so that must mean I know what I'm doing. No. So yeah, I'll speak to you guys later, and I might do some gaming in a second as well. So yeah, see you soon. Thanks.